Okay, if you don't mind, I don't have a microphone in this room, so I'm going to have to speak up a little bit. And it'd be great if I don't have to speak over anyone while we're going through the lecture. So we were running into a few technical glitches the first time out, so we're hoping it still works. I'd like to welcome you all back. Hopefully this is the third day of, no, second day? Second day, right? Second day of your semester, so I'm hoping it's been going well for you. I want this course to go well for you, and I'd like to welcome you back to your semester. I'm not sure how this is going to go. We'll see if it does actually play and if you can actually hear it. Any ideas how I can get this started, Jeff, at all? Or should we just skip it and just tell you it's a hilarious video? It's actually related to neuroscience. It's related to what I want you to do in this course as well. Because at the end of this video, what happens is, forget the guy's name, Sheldon. Sheldon's really tired. He's really cranky. I don't want you to be really tired or cranky in this course, and I'm hoping that throughout this course, you'll give me some feedback on how things are going. And I don't want this to be a course, even though we have a lot of content to cover, I don't want it to be a course where it's going to be overwhelming for you. So I'm going to take you through a few things related to what I think is important right now as geneticists, as well as in terms of neurogenomics. Right now, your field, in terms of neuroscience as well as genomics, is at the forefront of biotechnology as well as biology. There are a number of issues that many of you are aware of that's coming out right now in terms of the literature, in terms of neurogenomics. It is our discipline, what you're going to be learning in this class, that is going to be the most important in the next 5 to 10 years. It is not going to be neuroscience, nor is it going to be genetics. It is actually going to be neurogenomics that's going to be the most important field in the next 5 years. And those are things that I'm hoping to share with you throughout this course. So I'm going to take you through very quickly some of the different course material as well as the themes in this course. I'm going to unpack how you're going to be evaluated. It's your opportunity today to ask different questions, what sort of expectations I have for you, some suggestions for how to do well in this course. Again, it's really important that all of you do really well. In fact, we're going to go through an exercise on how to do well in this course on Thursday. We're meeting in a different room downstairs, but it will be a really short overview of how I hope you'll study for this course as well. I'm going to take you through some of the big issues in neurogenomics, but I'd also like some feedback in terms of what you think is really important in neurogenomics as well. Some of you are eating right now, and that's great. I want you to be able to come to class, to learn a lot, to enjoy coming to class, even though we're stuck in this marvelous Sid Smith room. Is anyone still somewhat hungry? Just curious. It's not a trick question. Are you guys lactose intolerant at all? No? You guys are okay with eating stuff that potentially contains dairy products? Yes? Okay, great. Blueberry or strawberry and rhubarb? Strawberry and rhubarb, anyone? If you're at all interested, please help yourselves. There is some yogurt there for you. I do have some hard copies as well. So if you need a copy of the notes because you're not registered in the class, I'll just leave these here. Okay. Does anyone need a copy of the notes? Okay, if I can just ask you to maybe circulate them backwards. Thank you for volunteering to, oh, more than one. Okay, okay. Is anyone else up for some yogurt? Yogurt is a good thing. I think we have a couple of spoons left.
Thank you very much, Dr.
Yes, no problem. Actually, it's not a trivial exercise. Yogurt is actually where some of our neurogenomics actually begins. Do you have any idea why I'm sharing yogurt with your classmates? It's actually not for making Danny feel more full or satiated. It's nothing to do with his hypothalamic neurons. It's actually to do with Genomics. Why are we interested in yogurt in this class? Yes. It could have something to do with the gut microbiome, and if we were only doing things related to HMB420, for example, yes. But in fact, no. Any other guesses? Gut microbiome, good guess, because what's in that yogurt? I can't believe you're licking the lid. Okay. Okay. Okay, that's how we roll here in HMB360. I like it. Okay. What is in that bacteria? It's Activia. So what kind of other stuff is in that bacteria in that yogurt? Anyone? So what else is in yogurt other than dairy products? We have what's living in there. So probiotic bacteria. So this is where our story actually begins. We are looking at today, we're going to be looking at how one technology actually originated from bacteria. When individuals who are involved in making cheese and yogurt, one of the things that happens is bacteriophages will actually infect bacteria. And when they do so, it turns the flavor of the yogurt as well as cheese off. And that's really bad. You can go through a whole batch, a whole vat of yogurt, and if the flavor is off because the bacteria have been infected via bacteriophage, it becomes really bad. And so one of the things that yogurt makers as well as cheese makers started to do was to investigate how to inoculate, how to protect bacteria from becoming infected from phages. And one of the things that they didn't realize is that inadvertently, one technology that they were using was known as CRISPR, because the form of CRISPR-Cas9 that we're going to talk about a lot in this course is actually a way for the bacteria to have an immune system against viral or phage attack. And we'll talk about this sort of as we go along today and a little bit more in detail in the next class. But I wanted to ask you, what else do you think is really hot and topical right now in terms of neurogenomics? You guys read a lot of papers, right? That's all you did during the break, just read papers and journal articles and review articles. What's really hot and topical in neuroscience or genetics right now? Anything? Yes, Sergeant? Okay, different types of mental disorders. Anything else? And definitely we are going to come back and talk about those. What else is really interesting? Yes. Optogenetics, absolutely. Anything else? Really interesting things that you would love to learn how to do or have read about? Yes. Okay, genetically modified rat models, all really interesting things. So one of the things that is really cool about neuroscience, optogenetics is now moving into human clinical trials. It's no longer just done in rats. It's going to be done in humans for chronic pain and others. So it's only taken 10 years from this basic science technique that was published in Nature to be working in a clinical trial in humans. So that's pretty amazing. With CRISPR, we're now taking a look at isolating different enhancers. Now, again, I know this is a neuro and genomics class. What do you know about enhancers, and why might that be important in the context of this course? Any ideas what an enhancer is, or why we might be looking at enhancers and knocking out enhancers to examine different aspects of biology? Yes. So they regulate gene expression, and why might we be interested in that? Okay. So we certainly are going to look at, starting today, turning on genes and turning off genes. But this is an enhancer which might regulate the function of different genes. So I want you to think about this throughout this course. Right now, in terms of neurogenomics, we have entered into a brand new era. So it's beyond sort of the era that we've all grown up with of understanding DNA and then RNA. So DNA now undergoes epigenetic modifications that we'll talk about. But now we also understand that so does RNA. And we also know that RNA can have epigenetic tags associated with it. And we also know that even though we have fully sequenced the genome, we haven't been successful in understanding, for example, why certain individuals have autism or reading disabilities or other types of neuro behavioral deficits. And we think now that it's a much more complex story than we ever thought before. It's not a single gene or genes that are responsible. It might actually be enhancer regions that are being targeted for these different types of behaviors. And we're going to talk about this, and we're going to be talking about the new technologies that are associated with this. So again, nowadays, we're required to tell you about whatever kind of financial or other types of conflicts we have. I have none. We don't have any financial or conflicts of interest in this course. I want you to be reading primary articles, but there is no textbook associated with this course. We're actually doing a reboot of 360. We decided to completely overhaul the course this year. So if you are okay with giving me feedback throughout the course, like I really hated that topic, please never cover that again in future classes. I'd love to hear about it. Actually, I probably wouldn't love to hear about it, but I'll still be interested in getting your feedback. But I want to really encourage you to think of this as your class. And I want you to really enjoy coming to class. I want you to really love neurogenomics. This really is the future of neuroscience as well as genetics. And I want you to be thinking of the fact that we are going to be learning about things in that sort of context throughout this course. So a little bit more about this course. So the material that you're going to get on your midterms and final exam is all going to be based on lectures. I will put the links for the primary papers or review articles or different things online that I find that are of interest for you, but you're not responsible for it. If I don't cover it in lecture, you're not responsible for knowing it on the final or on the midterm. The tutorials that we're going to have on Thursdays are there to really supplement or expand on the material or to provide you with an opportunity to do a presentation in front of your peers. We're also in those tutorials going to be involved in group work. You guys all love group work, right? It's your favorite thing of all time. You really love relying on someone else. You really love the fact that you've just texted your partner 15 times and they've never responded back. They ignore you and they have actually defriended you or unfriended you on Facebook. I know that you don't like group work. To be honest, I don't like group work. Not marking group work, but I don't personally like being involved in group work myself. Like even with other profs, there are profs that if I know that they're involved in a project, I'll actually stay away from that project. And so I want you to be thinking about this in the sense that why am I bothering to have group work in this particular course? I'll get to that in the next slide. I will, for every assignment, give you a review of what is required, and for the test as well as the exam, we'll also have an in-class review and reviews outside of the class time as well. So I want you to get really invested in the genetics, the genomics, the different technologies, as well as different aspects as they relate to neuroscience in this course. So I'm going to talk to you about a number of different new and emerging technologies, particularly related to RNA and RNA processing as we go along, as well as different applications. As I mentioned, most of you will have at least heard of and somewhat remember what optogenetics is. Not all of you have had a course in optogenetics. We'll go back and review this. But the way we use optogenetics now is very different than when it was originated 10 years ago. The field of optogenetics has actually moved on and is now into its 5th generation of optogenetics. I want you to be learning not the classic optogenetics, but the next generation of optogenetics in this course. I also want you to be thinking about how to do these applications from a developmental perspective all the way through to neurodegenerative diseases. We don't create these things for no particular reason. We create these different technologies because we want to be able to develop therapies, treatments, or to understand the disease process. And all of these different techniques, whether it's GWAS or optogenetics, or whether it's doing different forms of sequencing, are all important for us to be able to understand different types of diseases. As many of you know, right now, we are far outstripping our ability to do these types of experiments than to understand the consequences of these experiments. Some of you will have heard of some of the ethical issues that are associated with, for example, CRISPR-Cas9. Can anyone in 2 sentences tell me really quickly what some of the ethical issues related to CRISPR-Cas9 and genome editing are? Yes?
hierarchical system within human society simply by being able to edit the human genome. You could also have
sorry, 2 sentences. Okay, that's great. As many of you know, in some of my other courses, I like having like sentence cutoffs, and I'll get back to that a little bit later on. You're right. So is this going to be a freely accessible technology? Actually, the whole reason why scientists like CRISPR-Cas9 is it's so cheap. There's no other advantage. It's cheap. You can get an undergrad student doing this now for somewhere on the order of about $50 to $100 per experiment. That is actually cheaper than running like a Western blot. It's cheaper than doing staining. It's cheaper than sitting in front of a confocal. So it's incredibly cheap. But can we actually use genome editing? That's one of those big issues. Yes, Lauren. Okay, so editing out different types of diseases, and unfortunately in this course we're not going to cover that, but that becomes a big issue, and you're absolutely right. Some of you might think, wow, it'd be a great idea if we completely eliminated deafness, right? So there are forms of inherited deafness, and those individuals actually are very, very strongly opposed to that. They want their children to also not even have hearing aids or anything else, and those are issues that we'll cover. Frankly, from my personal perspective, I would love it if, for example, they created something that would grow hair. I don't know why, but I'm very invested in that type of technology. I wouldn't really mind, to be quite honest. But those are some of those big emerging ethical issues. And whether or not we can use these, because it's done now in the laboratory, it's been shown to be successful. There is a technique where CRISPR-Cas9 is used to create different organisms that have deficits associated with them. So they, in theory, can outbreed and eliminate certain species of mosquitoes, for example. Would that maybe be a good thing in areas where there are malaria? Right now, in my home country of Brazil, they have an issue where there is a Zika virus that's causing microcephaly. And maybe it'd be a good idea to eliminate the mosquitoes that carry Zika viruses as well. There are a number of issues, in other words, related to neurobiology. So we'll be covering things starting today related to CRISPR and Cas9. We'll talk about a number of different diseases and their potential treatments. We'll talk about emerging model systems that come out of all of this. So this is actually a really pretty picture that some of you will know. Does anyone have any idea over here on the right what this is actually showing? So our model systems are very different now, and they will continue to be very different than they were when I was in grad school and even when I was a postdoc. Probably by the time you reach the postdoc status, if you're going to go that route in research, you will have to know how to create these types of model organisms, different models. What is this? Any ideas? Again, no wrong answers at this stage. Yes? It certainly looks like a mouse fetus with different markers. There has been staining done on this tissue. What else do you think this might be? This is actually what is known as an organoid. This is an organ that's been grown from one cell, and that one cell was reprogrammed to actually form the entire brain. And this is a mini brain in a dish. It's not just hippocampal neurons, it's just not cortical neurons. There is a mini cerebellum down here, there's a mini hippocampus up here, and we can reprogram cells now using molecular, biological, and genomic techniques to actually create mini brains in a dish. And we can use CRISPR to actually go back in and see if we've created a mini Alzheimer's disease in a dish, can we see if we can come up with a way to eliminate some of the features of Alzheimer's disease or some of the proteins that are associated with Alzheimer's disease. So I'm going to again, take you through a few things in terms of unpacking this course, a little bit of your responsibilities for presentations, how you're going to be evaluated on the final exam, what sorts of things I want you to get out of all of the different types of assignments and assessments that you're going to have here. So Have all of you read the course syllabus? Yes, no, sort of. Some of you have, and again, I really encourage you to do so. Um, This is going to be only based on lecture material, as well as whatever I tell you is important in the tutorials throughout the course. I'd like you to attend lectures. We're going to try to stream as many of these lectures, provide you with the audio recording, give you whatever you want to have in this course. We'll try to provide that the best that we can. By the way, Jeff's been very gracious in terms of providing technical help. I'd love it if a couple more of you would volunteer to help out in terms of providing YouTube links wherever possible. So let me know during the break. It's only going to be multiple choice as well as short answer format on both the midterm as well as the final. The final exam will not be cumulative. I can tell you that if you've taken HMB 300, the neurobiology course, usually what happens is the midterm is fairly easy and the final exam is easy or hard? Hard. Okay, thanks for being generous. Harder. I can tell you it's reversed here. So what we're going to do is spend the first 3 to 4 weeks learning the different techniques, learning the different applications on the tests, etc. And I'm going to prepare you for that. And the midterm is actually, I would say it's about the same level as most of your first midterms will be. It's not going to be really, really hard. I'm expecting the average to be high. But the final exam is actually, I'm expecting it to be much easier than the midterm. And as many of you know, I don't believe in lying to my students. I want to be straightforward and honest with you about this. So the midterm is going to be challenging, but the final exam will be something that you will actually walk out somewhat enjoying. Not a lot, but you'll somewhat enjoy it, okay? So just to give you a little bit of a heads up. So I want you to hit the ground running, get these techniques under your belt, understand these techniques, learn to apply these techniques, and then be able to answer some practice questions that we're going to have for you. I can actually tell you ahead of time that in one of the tutorials before your midterm in class, you will actually have a number of questions. One of them will appear exactly the same way on your midterm, because we want you to get this practice and to start working together in groups. So we're going to start this Thursday. The room is downstairs, 1085. It's not in this room. I think it's 11 to 12 o'clock, so the timing is a little bit different. And most weeks it's either going to be a continuation of the lecture or it's going to be a supplementary method. I might expand a little bit on a different mouse model, for example, or I might tell you about zebrafish, or I might tell you about another technique that we didn't have time to cover in class. I'm also going to use this time to organize you into groups for presentation times later in the course after the midterm in March. You're going to be in groups. You're going to present together in front of your peers in smaller group settings, and we'll make sure that you're well prepared for this. There are going to be 2 times where the tutorials are going to be absolutely mandatory. I know that you guys are in 3rd and 4th year and you have a lot of things going on, but two of these Thursday tutorials, even though we're going to be providing you with different things throughout the course, 2 of them you're going to get 2.5 marks just for showing up. That's all you have to do. 2.5%, not even marks, 2.5% for showing up. You guys will all show up for that, right? I hope so. If you have a really good excuse, like you're really, really sick, or you're off to meet Fong Zhang somewhere for coffee and you're about to make a Nobel Prize discovery, that might be acceptable, maybe. But these two tutorial times are going to be absolutely mandatory. The one that's worth 2.5%, what we're going to do as a class, you're going to do this in groups, but we're going to retake the midterm without notes or anything else in groups, and you're going to submit again as groups the midterm again after discussing it, and you'll get a mark associated with that. So for example, if you got 85 on your midterm and you rewrote the whole test again sort of as a group, you submitted it as a group, everyone in the group gets the same mark, and now you've gone up to a 90, we're going to increase your mark on the midterm. And you're also going to get 2.5% for showing up to work in groups. Does that make sense to you? How many of you have taken, by the way, HMB 265? Okay, so it's similar to what you do in terms of quizzes. We're just going to do it 2 days after you've written the midterm. Do you have any questions about that? I'll explain it as we get a little bit closer to the due date. You'll write the midterm by yourself. 2 days later, you'll come into class. You won't have any notes. You won't have your laptops, but you'll get together in groups and you'll discuss what happened on the midterm and then fill it out again, and then you'll submit it again. You can't do any worse, by the way. So if, for example, you got an 85 on the midterm, your group decided to tank on you and decided that, yeah, you know what, I think it'd be a good idea if we all failed. Let's try to go for that magic number 40. You'll still get, as a group, the 85, okay? Like the lowest mark that you have is the lowest mark that you have, and we'll just make sure that you get a better mark after the group midterm. I'm hoping that no one actually does worse as a group on the second go around. And again, I want you to start studying wherever possible. Here's one of the reasons why I want you to get involved in group work. Did you have a question? Yes. Yes. Yeah, it's not going to be 50-50. Yeah, it's going to be a percentage of your midterm. So it'll probably be a split like 4-to-1. So you'll go up, definitely, and you'll go up enough to make it count, but you're not going to magically go from having like a 40 to magically having a 90. As fun as that might sound, okay? You got to study for both. Wait for the group? Yes, that's not going to happen. Yes. Let's just wait until that tutorial. I promise you, though, it's stuff that will be pretty much identical. You'll just be retaking this as a group, but you won't be, hopefully, going and bringing in your notes or your laptop or anything else. I know you'll talk about it on Facebook and other things like that. That's okay. Any other questions? You guys are looking at me like, sounds really sketchy. It's the way you'll have to wait and see how it actually goes. I'm more interested in you learning than in you actually the grade that you get out of it. As I said, the first midterm is application based, so it's a little bit more challenging than the final exam, which is less application based. And there will be some application questions, but it'll just be things that we learn about different techniques. Any other questions? No? So this is one of the reasons why I actually want you to be involved in group work. At some point in time, as one of my students, you're going to be asking me for a letter of recommendation, I hope. And I'd be very happy to write you a letter of recommendation. Many of these, whether you're applying for professional school or you're applying for graduate positions or you're applying even for volunteer work, now ask you questions related to your ability to work with other people. If I don't know if you can work with other people, I can't say anything about your ability to work with other people. I can't tell, for example, and this is actually from a recent reference letter that I wrote for someone, I can't tell if you're able to organize the efforts of other people. Can you actually organize a group, get people to show up on time, and then get them to do things as part of a group? I don't know, unless I actually have things that require group work. When that colleague of yours, that marvelous colleague of yours, has stopped responding to your text messages, what do you do? Are you able to actually adjust to this type of situation? Like, do you call up your prof? Hopefully not. I'm hoping that you don't have my number anyways, to be quite honest. You can always drop by the office, but what do you do when you have a new situation that requires you to respond appropriately? For sure, in this course, you're going to be asked, half of your mark is being able to work independently. The other half is related to how well you can work in groups. So find out who you're comfortable working with and spend the next couple of lecture times figuring out who you like to work with and who you feel that will really be a good team player that you're comfortable with. Not all of you are going to mesh well with other individuals, but find people that you're going to be comfortable with. It's your choice. I'm not going to be assigning you groups. If you know people in this class and you have worked with them before, great. Please work with them again. You can be in a small group, a group of 2. You can be in a group of 4, and it's your choice who you're going to be working with. I'm also going to be asking you to pick a recent primary paper somewhere between 2012 and 2016 related to a genetic technique that's used in neurodegeneration, neurodevelopment. It could be a behavior. It could be looking at, for example, musical ability and epigenetics. Great topic. Papers on it? Yes, not many, but there are papers on it. And maybe that's something that you want to be presenting. But I want you to be thinking about this throughout this course. You're going to pick a paper as a group, write a brief summary about it as a group, present it as a group, and then you're going to be writing a very brief, um, a brief sort of mini grant proposal that's going to be based on the one or two experiments that you would want to expand on based on that paper that you have just read. And we're going to go through this with you. There are some really easy ways for you to develop experimental questions based on published work, and I'll give you an example of that today in today's class. So along the way, if you're really worried about this, and please don't, I'm expecting you to do a fantastic job. As long as you can come up with 2 rational experiments and then come up with what you think might be the results in a rational way, you'll do fine. I doubt that anyone will do less than an A on that assignment, and we're going to have workshops related to this. You can always drop in and say, does this experiment make sense? My job is to help you along this process. There are only 70 of you in this class, which makes it really small, a really nice group of students to be able to work with. So we will talk about this group activity as this assignment after class as we get a little bit closer to this. So this is a reboot of a course. So that means that there is a lot of instability related to this. I'm a neurobiologist, so I always relate this back to something related to neuroscience. How many of you have taken PSL 300, which is like the best course ever, right? I'm not joking actually, I love PSL 300. I think it could be evaluated a little bit better, but the topics are really cool. Do you guys remember learning about the step cycle of locomotion? Yes? How you put one foot up, down, and it's repeated over and over again. Did you know that that mechanism is related to this dynamic instability? Your ability to walk actually requires a feed-forward mechanism. You don't have to think about where your next foot step is going to land. That's feed-forward, just a natural thing that you've been used to doing. So if you're used to studying in a course, you're using sort of a feed-forward mechanism. But what happens when you suddenly encounter something you haven't before? I've never done a mini sort of grant proposal. I've never had to do these types of assignments. Then you have to get a feedback mechanism in place. You have to be able to adapt quickly. And by doing so, this is one of the things that is my job. So the first part is yours. A feed-forward mechanism, you have to know how to be able to study the best that you can. If you are not succeeding well, if you're not succeeding well in terms of the midterm, then we have to figure out together how we can correct that. And we need this feedback mechanism sort of in place to be able for you to proceed. One of the things I'm going to ask you to do in every lecture that you attend, starting today, so the first part is just about how this course is going to be unpacked for you, is to engage in what I like to call 3x3. And this 3x3 is really simple. It's in your course syllabus. For every lecture that you attend, and in the second half of today's lecture, just remember one thing that really interested you. It might be like, yeah, I really like that picture of the dog. That dog was really exciting. I love golden retrievers, and I never knew that dogs were models for neurodegeneration or for developmental disorders. Think of one thing that is really challenging that you don't think you're going to have an easy time remembering. Like, I don't think I'm all that comfortable with exon skipping. And he mentioned it really quickly, but I know that that's going to be a problem for me in this course. So that's the second of the third. And then the third part is pick 3 slides, try to outguess me. Pick 3 slides that from the second half of today's lecture, because we're going to take it up on Thursday, that you think I would actually ask you questions on in the midterm. And we'll see if you are able to think just like me, because that's also part of my job, is to brainwash all of you into thinking just like me. And if I've done my job well, you will think just like me. And that means that you're going to do well on the final exam. So I'm your prof, you're my students, so I want you to communicate as much as you want to. Some of you are really shy about coming in for office hours. Some of you I can't see um, more than 2 or 3 times a week, and some of you I'll see like every single day. That's all fine. I don't really care. You're my students, and I take my job as your prof really seriously. And as I was posting on social media earlier today, I only have 3 courses, so I have a lot of spare time on my hands. So I actually like literally walk around campus looking for students to talk with. Why not save me the trouble? Drop by my office. Come and talk to me. I'm there most of the day. I only have like 1 or 2 hours of lecture every single day, and that's it. Like the rest of my time, I just sort of sit there, figuring out like who can I go bother. So, do you have any questions about any aspect of how you're going to be evaluated? Do you have any questions about any of the material? Do you have suggestions for any of the material that we're going to be covering in this course? Again, the key being it's your course. Any ideas, suggestions, things that you're not clear about, things that you want to talk about now? No? Okay, so if you don't mind, we're going to try to plow through a number of slides in the next little while. We'll take a break at 4 o'clock if that's okay. I'm going to try to let you out of here by 4:30 if that's not a big issue. Lots of really vigorous head nodding. Okay, I get the message. And we're going to start off with muscular dystrophy. You might think muscular dystrophy, not really that related to neuroscience, right? But actually, there are lots of things on this particular disorder that are actually related to neuroscience. Do you know anything before we get into today's lecture slides? Do you know anything about muscular dystrophy? What can you tell me off the tops of your heads? Yes. Okay, definitely genetic. Neurogenetics, neurogenomics. Yes.
Disorder which causes degeneration in a certain type of muscle. I can't remember.
Okay, so absolutely. It's an X-linked disorder that causes muscle degeneration in skeletal muscle, not just skeletal muscle, by the way, cardiac muscle, some smooth muscle is also affected. What else do you know about muscular dystrophy? Not on the slides, by the way. Yes. It is implicated in dystrophin, and we'll talk about that in a few moments. But beyond that, and not on these slides, and we will come back to this, many of these neurodegenerative disorders, and this is not a neurodegenerative disorder in the truest sense, this is a developmental disorder that we'll be talking about. Many of these developmental disorders actually affect the nervous system as well. Individuals that have muscular dystrophy also have things like autism. They also have developmental delays. They're not so quick to pick up on reading. They're not so quick to engage in conversation. So they look like they have autism-like behaviors in many cases, and this is very separate and distinct than the dystrophin that I'm going to be talking to you about. So is that something that we should be thinking about in this course, that these different developmental disorders, even though it's affecting muscle, also is implicated in many cases in the nervous system? Why is that the case? And why should we be looking at this? So as your colleagues have pointed out, muscular dystrophy is indeed genetic. It's an inherited disorder. It's progressive, meaning that if you've been diagnosed with it, depending on the form that you've been diagnosed, it is progressive. Much like Parkinson's, much like Alzheimer's, it gets steadily worse, which is what progressive means. You never go back, you never get better. So once you've been diagnosed with it, once the first signs and symptoms of muscular dystrophy come out, it's not going to get any better for you. It's going to be a slow and sometimes rapid downhill progression. This group of disorders as a whole, so there are many different subtypes, and I'll mention those in passing. This group of disorders will produce a loss of muscle tissue. The muscles will start to waste away. They will die off through a process of necrosis. There are different inherited types of muscular dystrophies, and depending on the type that you have, and the two that we'll be focusing on because they're actually linked and very, very similar in terms of their mechanism, both involving dystrophin, is Duchenne's muscular dystrophy as well as Beck's muscular dystrophy. Duchenne's, as I'll show you in a few moments, actually starts off in childhood, somewhere between the ages of 3 to 5. And Beck's is actually a slower progression. You get it in your early adulthood, and you can live a very long time with Beck's. You can live till like in your 60s, but the Duchenne's is very, very severe. It occurs in childhood, and when we talk about severe inherited disorders, that means it's lethal. So most individuals that have Duchenne's muscular dystrophy die by the time they turn 16, 17, or 18 years of age. Do you have any questions about that before we go on? So again, some of the symptoms, as you'd expect, if you have muscle wasting, especially in your skeletal muscles, some forms of this muscular dystrophy only affect some muscle groups, not others. Some are very, very specific for groups of muscles, as I'll show you in a few moments, while others can affect many groups at the same time. All of these symptoms have some severe consequences. So you have delayed motor skills as well as the usage of your limbs. Definitely, if your muscles are wasting away, you're not going to be able to use them appropriately. Some individuals with some forms of muscular dystrophy will have ptosis. And those of you who are in psychology, what is that? Yes. When it's like sagging, right? And especially around the eyelids. If you have a droopy eyelid, that is a very pronounced form of ptosis, where it kind of looks like it's not very active. If you see someone with a droopy eyelid, it could mean a lot of things. They could have suffered an acute disorder like a stroke, or they might have a muscle wasting disease like Duchenne's or other muscular dystrophies. And this ptosis is actually fairly common in a number of different neurodegenerative and developmental disorders. One of the things that very typically happens is a lack of coordination as the disease progresses. And of course, the one thing that is almost common among all forms of muscular dystrophies are the loss of muscle size and the accompanying strength of the muscles as well. Eventually, these individuals can't walk. They're not able to stand. You'll see them in crutches, then a wheelchair, and eventually they'll be bedridden before they die. So again, the two types that we're going to be focusing on is Duchenne's muscular dystrophy as well as Beck's or Becker's muscular dystrophy. There are many, many other types, myotonic, Steinart's, as well as facioscapulohumeral, and these are all targeting specific muscle groups. Okay, and we'll talk very briefly about some of these others. Now, you don't actually have, and there's some suggestion that there might be up to 20 different types of muscular dystrophies. Now, how would you categorize them? How would you classify them? Any ideas? You have 20 different types. How might you start classifying these? Yes. So by the types of muscles that get targeted? Definitely, yes. Other ideas? Age of onset is another way we do this. Yes. Severity can be another way we do this. Arslan? The genes that are affected is another way, and also on the pattern of inheritance. Some are X-linked and some are autosomal dominant. There are many different forms. Yes? Speed of progression, definitely. So you don't have this diagram. It's just to demonstrate this for you. I'm not going to ever ask you a question on a slide that you don't have in your notes. Hopefully, if this is streamed and you're really interested, this will come up for you. This is an example of the muscle groups that are targeted in Duchenne's as well as Becker's types of muscular dystrophies. This is the Emery-Dreifuss type over here. This is the limb-girdle types. And you can see that different muscle groups, some affecting the eyes, for example, in the oculopharyngeal type of muscular dystrophy, your eyes have ptosis. You can't really use your eye muscles well. Many, many different specific types of muscular dystrophies inherited in different ways. And again, there's some suggestion that we might have up to 20 of these different types of muscular dystrophies, different speeds of progression, different ages of onset, different genes that are being affected, etc. Do you have any questions before I go on? And all of you have taken at least one physiology course, right? No matter if it's animal physiology or human physiology? Yes? Okay. So again, when we look at Duchenne's, and there's a rationale for why we're going to be looking at Duchenne's, this accounts for more than half of the different types of the muscular dystrophies. Of the 20, the vast majority, half of them, if you see someone with a muscular dystrophy, it's usually due to Duchenne's muscular dystrophy. And again, it's X-linked, which means that the mother is usually the carrier. The mother is usually not affected. They don't have problems with their muscles at all. They don't have any of the overt signs unless it's a very severe form of muscular dystrophy. But it means that the males are going to be affected. Okay, and because it's carried on the X chromosome, which is affected, usually you have a son associated with muscular dystrophy being that X-linked form of inheritance. So again, the female carriers tend to have very, very mild symptoms, whereas both Duchenne's as well as Becker's muscular dystrophy affects boys. The onset is usually earlier for Duchenne's muscular dystrophy, usually at the age of 3 to 5. So again, think about that, the age of 3 to 5. What are you doing at the ages of 3 to 5? Sorry? So you're walking, running around, yes, other things. You're undergoing some of your first growth spurts, anything else that's going on. It's a very important developmental age. And of course, suddenly, you've started to lose motor coordination. You've been progressing fine. And then at the age of 3, you get the onset of this muscular dystrophy. It can be devastating for a family, as you would well imagine. So again, this tends to be very, very rapid. It is a lethal disorder. It's usually death in the teenage years. Once you've been diagnosed with it, you never get better. Your muscles start to waste away. And in about 1/3 of individuals, even though we know that it is an X-linked inherited disorder, in 1/3 of individuals, as far as we can determine in the literature, 1/3 of individuals that have molecular diagnosis of Duchenne's muscular dystrophy, so they have a lack of dystrophin in their muscles. 1/3 of those individuals have no genetic inheritance. Their mothers are fine. They don't carry the any issues in their genes for Duchenne's. So that means that this is a spontaneously arising mutation. This happens to be in one of those hotspot regions. This is a very, very large gene. And in this hotspot region, 1/3 of individuals that have Duchenne's, where they have no dystrophin or truncated forms of dystrophin or other manifestations of dystrophin, they have spontaneous mutations. We don't really understand it. We are going to come back and circle back to how these spontaneous mutations can occur, but I wanted you to be aware of the fact that it doesn't always have to be X-linked as a form of inheritance for Duchenne's muscular dystrophy. And again, the muscles in the biceps in an individual with muscular dystrophy will start to shrink and atrophy. Have any of you seen a clinical presentation of muscular dystrophy in a young individual? So you can imagine their leg muscles getting thinner and thinner, their calf muscles getting thinner and thinner. Before that happens, do you know what occurs in those individuals? They actually have an expansion of their muscles. Now, I want you to think a little bit about that during the break. We'll come back and we'll talk about it, then we'll go through the remaining slides. It's not on the slides, and it's something I want you to be that I want you to be aware of, and you'll need to know it for your midterm. But I'm going to ask you during after the break to let me know why you think that initially, before these muscles start to atrophy, that in fact they get bigger. And tell me what you know about the physiology of muscle tissue that might allow for that kind of phenotypic expression. And we'll take a break now. We'll come back and we'll talk a little bit more about Duchenne's. One of the things I had mentioned before the break was that this is sort of a reboot of the course from last year. And so we'll be using like a new format in terms of testing and everything else. How many of you feel uncomfortable if, for example, you don't have like previous tests or previous exams? Really, not so bad. Well, you guys are a very chill bunch. Okay. How many of you would feel not so bad if you had a few questions that would be like a good practice for you before like your midterm in terms of both multiple choice as well as short answer? Okay, so we'll try to arrange that for you. Here's an example of a slide that I don't want you to actually memorize. So if you're thinking you have to memorize this slide here, please don't. I don't want you to memorize the particular order of the different truncations or anything else. It's just to give you an idea. There are some elements of this slide that I'm going to point out that I do want you to know. And in terms of genetics, we can always really be nitpicky and ask you about specific genes and their loci, but wherever possible, I'm going to tell you what you should be concentrating on this particular slide. So when we're talking about Duchenne's and Becker's muscular dystrophy, we're talking about the X chromosome, and we're talking about the P arm of the X chromosome on locus 21. And on locus 21, which has this large stretch, this is a really large gene, about 2.5 megabases, which is a huge stretch of DNA. We have a number of different exons. In fact, this particular gene has 76 exons that you don't see here, and you don't have to worry about all of these details. But I wanted to point this out to you. Dystrophin itself is a massive protein. How many of you have run Western blots? You've done Western blots in your research. What's the size of your protein? Yes, everyone look away who put up their hand in shame because you don't know the size of your own protein. What's a normal? Is 50 kilodaltons a big protein? Is 100 kilodaltons a big protein? 180, 200, those are pretty big size proteins. Those are structural proteins. This one is 400 kilodaltons. This is massive. This is a big, humongous protein. It is required for structural stability, as I'll show you later. This is the gene that's being disrupted. So this gene forms this big, massive protein, but there are some issues with it. And we won't go through all of this right now. In some cases, we don't actually form the gene product that you see down here on the bottom. We don't actually form dystrophin, and we end up getting what is known as Duchenne's muscular dystrophy. Duchenne's, we don't have as much dystrophin as we normally should have. In some instances, like Becker's type of muscular dystrophy, we get some formation of dystrophin, but it's truncated, it's shorter, it's not as long as it should be. It's not that massive 400 plus kilodaltons. We get these little revertant types of dystrophin. So we can, in theory, form these truncated, smaller versions, but in Duchenne's muscular dystrophy, we have almost none. In our muscles, in muscles of an individual with Duchenne's, they have none of this gene product known as dystrophin, and they have very small amounts of these truncated forms of dystrophin that we'll talk about in some more detail. So that means their muscles don't have this protein. That means that behaviorally, they have these issues with their muscles. One of the characteristic clinical features of Duchenne's muscular dystrophy is Gower sign. Again, it strikes up between the ages of 3 to 5 typically. This is Duchenne's. We don't see this in Becker's type, nor do we see this in the other types. This is where an individual has been progressing normally, and now at maybe the age of 4, can no longer stand up properly by themselves. And you can see sort of diagrammatically on the left, as well as on the right, they actually have a number of issues. Their hip muscles have weakened. They no longer produce dystrophin. The muscles aren't bulky. They're no longer able to contract. And as a result, when they try to stand up, they have to assist themselves as much as possible. And the way that they stand up is known as Gower sign, as you see here. So this is diagrammatically here on the left. And this is a young boy that you see here trying to stand up. He'll have to roll over onto his stomach. And then from his stomach, he'll start walking up his own limbs. That's Gower sign, walking, using your hands to walk up your own limbs before you're able to stand upright, because his muscles have been affected, particularly the muscles in the leg as well as around the waist. So how would you diagnose for Duchenne's muscular dystrophy? By the way, how many of you think that this might be one of your 3 slides that you want to circle to memorize? So 2 of you, those, yes, this slide, not, yes, this immediate slide. I think I'd have a really hard time actually trying to write a really cool short answer question related to this. I could be really, yes, I'm hoping you don't forget about Gowers, but this is probably a good slide for you to make sure that you know. And again, it's going to be application based. So not just knowing this is enough, not memorizing this is enough, but applying this in a logical way will be beneficial for you on your midterm. So Gower sign is a compensation. It's the way that the individual walks up their own limbs to be able to stand up, and it's a compensation for the hip muscles as well as the upper leg muscles starting to atrophy. Their muscles are starting to weaken. They're disappearing. They're gradually going to vanish altogether. If we were to take a look at that individual who exhibits Gower sign, who probably has Duchenne's muscular dystrophy, They will also have elevated levels of creatine phosphokinase, or CPK. And often we see this denoted as CPK-MM. So this is a genetics class. This is a genomics class. What does the MM stand for these types of enzymes? So enzymes, there's only one type, right? Yes, no? So we have isoforms of these enzymes. We have enzymes, creatine phosphokinase that's found in the heart, and we also have creatine phosphokinase that's found in the muscle. And this stands for one of the muscle types. So when we see an elevation of creatine phosphokinase in a young individual in their blood, we know that there is an issue with their skeletal muscle. We could also go in and do a muscle biopsy and find out that they don't have muscle dystrophin. When we punch out a little section of their muscle, we will find, and when we run it by protein analysis or we use mass spec, we would see that their levels of dystrophin, as we'd expect, would be very low or completely absent in these individuals that are expressing things like Gower signs. We could do a genetic test, and we'll go into this in more detail in coming lectures, for XP21, the X chromosome, the P arm of the X chromosome at locus 21. And we might also start to examine whether or not it's not just their skeletal muscles, because many of these individuals, as I said, their skeletal muscles are definitely affected. Skeletal muscles have lots of dystrophin, normal skeletal muscle, but they might also have arrhythmias. They might have cardiomyopathy. They have heart problems, in other words, in addition to that. Do any of you know what CHF is? In physiology class, have you covered that at all? Congestive heart failure. So they have issues where their heart isn't functioning well. They're not able to pump blood as efficiently. It starts to back up on one side in the pulmonary circulation, so they have a hard time breathing. And of course, the muscle mass is going to decrease as the disease progresses. But as I mentioned before the break, one of the things that happens early on in those young individuals is, in fact, their calf muscles become really, really large. Why? What's happening to their muscles before it starts to atrophy, before it starts to die by necrosis? What do you know about muscle? Yes.
Because dystrophin forms a structure
Yes.
so that the muscle gets damaged more easily.
Yes.
So when the muscle gets damaged more easily, it gets repaired in the same way that when you're trying to like bulk up like bodybuilder kind of thing.
You know, I want to say something about lifting. I'm going to avoid that for the time being. You're absolutely right. When you are trying to build up your muscles, what you're actually doing is damaging your muscle to bulk it up. But it bulks up in what way? How does it actually bulk up? Yes.
Is it replaced by satellite cells?
Satellite cells. These satellite cells, which are at the periphery of the myofibril, come in where there is damage. So their muscle is damaged. They have necrosis. So their satellite cells actually come in and try to make use of the fact that there's damage there, and they'll start to divide like crazy, and they'll compensate for a while. The muscle looks bigger, it bulks up before it starts to atrophy. So that's great. I'm glad that we have a very well-read class that knows a lot about physiology. Even though it's not on this slide, I'm going to ask you to know that one type of cell that we see in muscle fiber is the satellite cell, and that compensates for this original atrophy in Duchenne's muscular dystrophy. Again, as I mentioned to you before, in terms of the molecular diagnostics for Duchenne's muscular dystrophy, dystrophin, which is encoded by the XP21 locus, is the largest known human gene that we have. It's about 2.5 mega base pairs, which is enormous. It has 76 different exons, and it encodes for a very, very large 427 kilodalton protein, which makes it a massive protein. Normally, it wouldn't even migrate into the gel. So this codes for a large protein that's found in skeletal muscle. It's absent in Duchenne's muscular dystrophy. It's also encodes for dystrophin that's found in cardiac muscle, explaining some of the issues with cardiomyopathy and others. It is also found in some other forms of smooth muscle. It's also found in other places as well. So a number of different systems get affected. It's not just skeletal muscle. Why is this all important? Dystrophin acts sort of as a bridge between the extracellular matrix as well as the actin fibers inside the muscle. And this allows for the appropriate connection as the muscle starts to contract. Now, I don't need you to memorize this diagram down here. I'm putting it up here because as a genetics and genomics class, we will eventually be talking about promoters. And you can see that for dystrophin, there are 3 different types of promoters that cause different types of expression in different tissue types. Dystrophin is expressed using the brain promoter in brain tissue. It's expressed in muscle tissue using the muscle-specific promoter, and it's also found in Purkinje cells in the cerebellum. Again, all of that extraneous detail you don't need to know. It's just to reorient you a little bit with some of the nomenclature that we're going to use and some of the diagrams that we'll be using. You will not see this diagram, nor will you be responsible for this diagram or any information, nor even the promoters that I just spoke to you about a second ago. But this is an important slide for you to know. And you don't need to know all of the details here. And you don't need to memorize all of the different structural elements that you see here. In fact, I couldn't tell you without looking at it all of the elements that are found between the sarcolemma and the extracellular matrix. But this is this massive protein that we're looking at. This is gone in Duchenne's muscular dystrophy, and it's smaller in Becker's muscular dystrophy. And you can see that it helps to bind and organize a number of different complexes that sit at the level of the sarcolemma. And I'm going to ask you here now, what do you remember about the sarcolemma? What is that? Sarcolemma. Yes.
The cross bridge between the actin and myosin.
Almost, yes. We definitely have actin filaments down here, which means that interspersed we would have myosin. But what is the sarcolemma? Yes.
It's a calcium storage.
Almost. The calcium storage is intracellular. That's a sarcoplasmic reticulum. But this is on the outside. What is the sarcolemma? Yes. Is it? Those are the T tubules, I think, that you're talking about. You guys said that you took physiology. I forgot to do the follow-up question. Did you all do well in physiology? The sarcolemma is the plasma membrane surrounding the muscle cell, right? It's just a fancy name for the plasma membrane around muscle cells. And the sarcolemma will depolarize, it will start to activate these actin filaments, but this dystrophin acts sort of as a bridge. It acts to tell the muscle when it's contracting and when it should be contracting, and it also helps to protect these actin filaments. Now, I don't need you to memorize all of the details that are associated with it. Interestingly, in many forms of muscular dystrophy, we don't really see mutations or truncations in any of these other proteins. It tends to target the ones that are associated with actin. And we'll get back to that a little bit later, but you don't have to memorize these elements on this diagram at all. Okay, it's just to give you an overview of what dystrophin is, that it binds to many different proteins that link from the extracellular region at the top, the extracellular matrix. It spans, that complex spans the sarcolemma, which is the membrane that surrounds muscle tissue, and it helps to cover up these actin filaments. And again, this is shown to you in another way. And the major point that I want you to take from all of this is this massive dystrophin protein up here at the top has actin binding regions. It's massive. It's 427 kilodaltons in length. There are a number of structural elements to it. And what happens in a normal individual, it will encode for every last one of these 76 exons. But in Becker's, where there is a large deletion, many portions of these rod domains are gone or missing, the reading frame is left intact. So in other words, it gets shorter, but for the most part, it can still do its job. It's just a shorter miniature version of what dystrophin should normally look like. So it can still interact. It can still interact through the C terminals with many of the proteins that you see here. It's just not as long as you see it in this particular diagram. This is different than in Duchenne's. In particularly severe forms of Duchenne's muscular dystrophy, there will be a very small deletion. Not many of these exons, maybe only in one exon, which is deleted, but the entire reading frame gets changed. That means that we don't form dystrophin, a lot of dystrophin, or the forms that we do have tend to be very aberrant forms of dystrophin. So because it's used as an anchor to the extracellular matrix, and because it's working to communicate contraction as well as how it should be bound to the extracellular matrix, this dystrophin plays a huge role. And as a result, if you don't have dystrophin, every time your muscle contracts, and there is no bridge between muscle contraction, action potentials on the sarcolemma, actin cross bridges occurring, this muscle contraction now, in the absence of dystrophin, actually stresses the plasma membrane, the sarcolemma that I mentioned to you before, causing damage in those myofibrils, those actin myosin containing fibers, and eventually it will cause them to undergo necrosis and cell death. That's why the muscle mass decreases. They die. They die by necrosis. Now, again, I don't need you to memorize all of these different diagrams. Some of you who are in cell biology will have seen this diagram multiple times. There are certain elements, though, on this diagram that I want you to make sure that you know and that you go back and review for your midterm. And I will tell you in advance that this is one of those 3 slides that I myself would pick, and it is a slide that I would use to generate a multiple choice test question on. So this is what the normal sarcoplasmic the normal sarcolemma, what it looks like. This is the normal sarcoplasmic reticulum. Skeletal muscle has an internal store of calcium. It gets released from this sarcoplasmic reticulum. Here's that dystrophin, which is covering up the actin, and it's normally doing its job. However, in the absence of this dystrophin, every time there is a calcium influx, because everything else is still there, every time there is a calcium influx, this large increase in calcium that goes above and beyond what we would normally see actually starts to do a number of things that is deleterious for this particular cell. So it will initiate, and this becomes important in a few moments, an inflammation cascade, the NNOS, the NF kappa B, the INOS, and the inflammation cascade. This excess calcium that is now resulting without having this dystrophin here will actually activate calcium-dependent proteases, one of which is calpain, which loves to digest different intracellular proteins. It will also dysregulate the mitochondria. It will increase intracellular levels of pKa as well as CAMK2, causing aberrant phosphorylation of the sarcolemma, the sarcoplasmic reticulum containing channels like the ryanodine receptor here, the RYR, causing more calcium to come out of the sarcoplasmic reticulum. So all of this is sort of a positive feedback mechanism. Without dystrophin, we have way too much calcium. Way too much calcium actually causes cell death in a number of ways. One, through mitochondria, activation of calpain, more calcium in a positive feedback loop through the sarcoplasmic reticulum, etc. Do you have any questions about this before we go on? Any ideas? Suggestions? No? So I want to actually start you thinking. And there are elements that I want you to remember from the remaining few slides, but I want you to start thinking about this. In our discipline in neurogenomics and neuroscience and genetics, one of the things that we often use are different model systems. And I told you about one, an organoid culture system that we can use, a mini brain in a dish, for example. But oftentimes we use animal models. And are animal models really all that good? Sometimes they are. Sometimes they're fantastic animal models. We'll talk about, for example, making mice smarter. All you have to do is introduce human glial cells, and these mice become smarter. We don't understand the reasons why. We don't know why it's happening, but we can use animal models to look for things like intelligence or for memory and other things. But when we're looking at dystrophin and we're looking at muscular dystrophy, how do we start looking at this? Well, the obvious thing would be to take muscle biopsies. Let's figure out what's going wrong in the muscles of individuals that have muscular dystrophy. Taking these repeated biopsies for research purposes, not for diagnostics, because you want to do that to diagnose this 100%, is actually unethical. To get a young child to continually give samples of their muscle, which is painful. A biopsy is where you're literally punching out a section of the muscle, is actually unethical. Our technology is rapidly increasing, but we don't have non-invasive imaging technologies in muscle that are at the level that we can image molecularly and even at the cellular level quite yet. So one day we'll get to that stage, but right now we're limited by the fact that we don't have this. So there are other models that we'll be talking about other than the one that I'm going to show you, the MDX model. There are indeed dystrophin deficient models such as the golden retriever muscular dystrophy model. You need to know that as an animal model. I don't have all the details up there. I do have a cute picture. Well, it's not so cute actually. I have a picture of a dog and two dogs on the next slide. But can you think of a reason why we don't use golden retrievers or retrievers in animal research? We do up to a certain point. Why wouldn't you use dogs? Yes. So certainly they are really nice, higher order, much more higher order than say mice for sure. Anything else? Yep.
They have a longer reproductive cycle.
Longer reproductive cycle, takes longer to breed them. Definitely. Anything else that you can think of? Yes. So do mice. Unless you're talking about Pinky and the Brain. Yes. Anything else? Not bad though. I like the fact that you're thinking about this. Yes.
Stress affects animals in different ways, so maybe it's easier to kind of anesthetize a mouse than a dog. Well, it's probably easier to anesthetize a mouse than a dog.
Okay, so being able to manipulate these animals by anesthetics, etc., might be easier in smaller versus larger animals. Yes. More expensive. Anything else? Yes. So that again was mentioned. So bipedalism in mice versus dogs, it's a good idea. It's a great thought. I like that thought. But we do use mice, and those are not bipedal. But that is a good consideration as well. Yes.
Kind of goes off the guy who said they have a long reproductive cycle.
Yeah.
Maybe it's harder to use something like CRISPR to develop a really good genetic model.
So developing a good genetic model using CRISPR because of their longer reproductive cycle might become an issue as well. All great ideas. Every last one of those were great ideas. And yes, there's something to all of that. Bipedalism versus forelegs, etc. But it really comes down to that one issue that one of you pointed out. It's cost. Dogs are much more expensive to house as well as to invest in than mice. We do use these. If we think we have a good treatment strategy developed for mice, we will then take it to dogs. We don't take it to primates, we take it to the golden retriever. That's another issue that we'll be talking about in this course. Now, we see severe muscle wasting. On the left of that dog, we also have a lack of coordination, muscle wasting on the dog on the right as well. Dogs, because of the genetics and the breeding of certain dog strains, like the golden retriever, they are prone to develop muscular dystrophy. Certain breeds have been shown in the literature to actually develop full-blown muscular dystrophy, including the wasting away of muscle in their legs, etc. But the most common model, and the one that I'm going to be introducing to you, is the MDX mouse model. It is the most commonly cited, the most commonly used model, the one that I want to spend some time going over with you. Just like we have mouse models for different neurodegenerative conditions, here we have a mouse model that we can use to our advantage over the last 30 years or so to be able to model muscular dystrophy. It results from a premature stop codon. It was a spontaneously occurring mutation that was observed, and it was a premature stop codon that was introduced into the MDX, so very similar to different forms of muscular dystrophy. It results in a truncated dystrophin, which is terminated after exon 23, and again, their dystrophin is very similar to our gene. It was also found in the C57BL6J background, which is the type of mice that we normally use in research. Danny.
If it's a truncated dystrophin, wouldn't it look more like vectors than MDM?
Ah, okay. I'll get to that in a second. It's a great thought. Would it look like Duchenne's or would it look more like Becker's? It's a great thought. And I want you to be criticizing and coming up with these types of criticisms throughout this course. So in MDX mice, before I get to that point, mechanical damage produces a loss of functional myofibers. They atrophy, just like we would expect to see in Duchenne's. They also appear to be more susceptible to contraction and stress-induced damage, resulting in sarcolemmic tearing, resulting in atrophy and necrosis, etc. But there are issues with this. So there is also physiological control lost of calcium homeostasis. They've also been reported to have elevated levels of NF-kappa B, which I just showed you is responsible for inflammation. In human Duchenne's muscular dystrophy, one of the other things that we see as the disease progresses is we also see elevated levels of inflammation, including things like having NF-kappa B levels being elevated. But this gets back to one of the things that Danny had pointed out, and many of you are probably thinking as well. There are some issues with this mice. It's not a perfect animal model system. So one of the things that is different, I told you earlier on, Duchenne's muscular dystrophy, it's progressive. Usually by the time they start, they die in their teenage years. They have a normal lifespan. These mice live just as long as their wild part C57 counterparts. It is not a perfect model. They also don't have a huge loss of muscle function. They are still able to walk around. They're coordinated. They live just as long. And one of the reasons why this happens is not only for the reason that Danny pointed out, that yes, it's a truncated version. There's still some dystrophin there. In fact, one of the things that we see is that there is a related molecule, utrophin, which I'll show you in the next diagram, which has 80% homology. Many of the same regions, it's just a shorter version, actually gets upregulated. And it can functionally replace the dystrophin and interact with the same network of proteins. This is utrophin. Again, don't memorize the diagrams at the top. Don't even bother with the diagram down here. It's just to show you that overall, it looks a lot like dystrophin. It's just a shorter version. And yes, they still produce truncated versions of dystrophin as well, but they also overexpress now utrophin when dystrophin is lost. So it's not a perfect model, but it is the model that we're going to be looking at in this course. And you can see that it still interacts with actin. And if you're wondering what this ABD is, it's an actin binding domain 1, actin binding domain 2, because it interacts with actin just like dystrophin does here at the top. Interacts in a slightly different way, but it can functionally replace dystrophin. They live long and prosper. Sorry, I had to say that one. So people were wondering, okay, we have this spontaneously occurring mutation. It's well established in the literature. We've got 30 years of research. Do we just throw all of that out the window? How can we make this a better model system? So these are improvements and refinements to the MDX model system. One is, if you're contracting muscle and that causes them to get lost, why not try to make those muscles contract even more? Put them on a forced exercise. So that's what some researchers do. They speed up the process. Make them exercise a lot more damage, more loss, less calcium homeostasis. And these forced exercise models would be one thing that you could do. How would you know that you are successful, by the way? Say I asked you that on a multiple choice test question. How would you know if this is actually successful? What might you show or look for to see if this is a good model for Duchenne's muscular dystrophy? Yes? We could look for satellite cells, might be one possibility. What else could I maybe look at in the blood or elsewhere? Yes?
In the blood, you could look at the creatine.
So we could look for CPK, absolutely. What else is a diagnostic feature of Duchenne's? Yeah.
Can't you look at it behaviorally? Can't you just see the
Yeah, possibly.
of the mouse
Sure.
deteriorating at a rate faster than the control?
Yep, absolutely. We could look for muscle atrophy. Yes?
Exercise, you can't expect muscles to grow. Maybe instead of seeing them shrink, actually be
Okay, so maybe they get smaller with increased exercise and increasing the level of atrophy. All things to be thinking about. This is an example of an application type question, not rote memorization. Taking what you've learned about Duchenne's, applying it. Like, is this actually a better model? How would you show it as an application question? I want you to be thinking about those. And as many of you know from my other courses, that's the way I like to ask multiple choice test questions. There is an MDX52 knockout mouse. all of those revertant forms that Danny had mentioned, the truncated, smaller versions that still exist that might compensate for not having the full-length dystrophin. In the MDX52 knockout mouse, all of those revertant forms of dystrophin are gone. There's no dystrophin at all, period. And that is a much better mouse model system that goes along with this. By the way, in this forced exercise model, one of the biggest issues is everyone gets different results. In science, we want standardized results, but everyone has a different exercise protocol. They keep them on there for half an hour. They keep them on there overnight. They see just a 10% increase in CPK. Another group says they get like a 30%. So these varying protocols for the forced exercise makes for a large amount of variation in the resulting damage and the efficacy of the model overall. Another model system that you might come up with, anything that you can think of? Yes?
So none of the models that are referenced now that address utrophin at all. Like they're just letting
Yep.
they just say it's compensating. We're just gonna leave it be, kind of thing.
Yep, pretty much, yes. They're not worried at all about utrophin.
But isn't it allow them to live a healthy, irrelevant life?
So that's a great question. So how would you answer that? So one of the things that you might look with Duchenne's muscular dystrophy, what would you look for?
Well, they die early.
Yeah.
So if the mouse is able to make it to a relatively comparable life compared to a mouse that isn't affected by
Yep.
this knockout,
Yep.
then there's the discrepancy between the.
So some discrepancy.
It needs to die at one point earlier than it would if you didn't do it.
Sure, I think that's a logical approach. One of the other things that scientists look for in Duchenne's is in humans, what happens to utrophin? Does it also go up? In fact, it goes up for a little while, but overall, we don't see that compensation in human Duchenne's muscular dystrophy. So why am I bothering to mention all of this? This is one of the coolest things in science. And in science, it's very, very rare. You see this once in a while, where you get a back-to-back paper. You get 2 articles in the same issue of a top-tier journal like Nature or Nature Neuroscience or Nature Genetics. Here, over the holiday break, right at the end of the year, before 2016, right at the very last day, at the very last hour that you could possibly upload something online, 3 articles came out back to back to back. And it was a tour de force because it really was, it was sort of a backhanded way for the editors of Science to say, look, this is the technique of the year, the molecular biological technique of the year, and this is what is going to drive all research in 2016. And that was their way of saying, look at the strength of this technique that is now pervasive in science. So if I were to ask undergraduate research students who are doing projects in undergrad, because I do fairly regularly, how many of you use CRISPR-Cas9? Actually, you'd be surprised, but I do get students that are using this right now as undergrad, 3rd year and 4th year undergrad research project students. It's that easy to be able to use. But I want you to think about it this way. This is a back-to-back-to-back paper. 3 papers that came out in Science in the same issue. All of them showed exactly the same results. I am going to go through some of those results with you in the upcoming week, next Tuesday. But I want you to be excited about this discipline because this is sort of the signal that this is the brand new start of a brand new era with the most powerful technique that we've been able to develop, which if you are listening to the pundits that say that if this is allowed to go unchecked, this will, in theory, wipe out the human race, in theory, within 3 generations if it's allowed to proceed. We have 3 generations because we have a very long reproductive cycle. But in fact, they're suggesting that without the bioethical standards in place, that because this technique, if you're looking, do you see any commonalities in these 3 articles? What jumps out at you? Yes. Postnatal. It's in adults. That's number 1. Yes. It's in a live organism, an adult live organism. What else? Yes. Sorry? They are also targeting muscle stem cells. All of them are showing exactly this, that we can alter the the animal genome in this mouse model, have functional recovery in the adult, and we can do this on a reproducible level. And there are fewer of these off-targeting effects. The other thing that, as you get a little bit more into reading papers, if you start looking at the names, there are a couple of names that hopefully some of you will recognize. Do you recognize any of those names? Mine's not up there in case you're wondering. Any names that you recognize? Yeah, so Fang Zhang is actually on 2. It's nice to be the leader in your field. So Fang Zhang is right there. He's also right here, back to back. Anything else? Yes. Yep, absolutely. He's definitely up there. Do any of you remember Amy Wagers? Those of you who took HMB 300 with me this past year, this past semester. Have to go back and revisit your grades, maybe think of lowering your grades a little bit. Amy Wagers, Harvard University. Do you remember her at all? Controversy? GDF 11? Parabionts, exchanging blood from young to old animals. So she's been very interested in factors that are in the blood that can influence health and disease and regeneration. And this is one of the reasons why she is looking at these dystrophic mice. So Amy Wagers is a huge name in terms of muscle regeneration, aging, and aging factors. You might think that this is just out of the blue, but do any of you know Eric Olson at all? Not on a personal, like, hey, Eric, how are things going? Shall we go out for a drink sometime? But have you heard of Eric Olson? So this looks like it was a tour de force. But just one thing to consider. If you go back in the literature, and the first author is the same first author. This was published at the very tail end of 2015. He had actually published a paper at the beginning of 2015 that was looking at going in and editing the genome of fetal mice to completely eliminate muscular dystrophy. So he had actually been involved in this for a while, and he was able to do this whole experiment in a year and publish it in a year. Now, can you imagine having a technique that allows you to be able to do all of this within the span of a year and published in a journal like Science? And this is really why I want you to be excited and interested in this technology, because whether you think so or not, whether you think it's all pie in the sky, this really is proceeding at a much faster pace than anyone had ever imagined would occur. And the fact that 2 weeks later, also in a high impact journal, they were able to reverse blindness in mice using CRISPR-Cas9 is pretty remarkable in the sense that we can now target different neurodegenerative conditions as well as different developmental disorders. This is a developmental disorder. These mice had Duchenne's muscle wasting. They're not perfect models, but these animals functionally recovered as adults. Now, could you imagine offering that hope to someone who's a teenager who's looking at 2 years of life and potentially dying in 2 years with this type of CRISPR technology? So we'll sort of stop here. I have to apologize that it went on a little bit longer than I had intended. So here are things that we're going to be looking at down the road. We already have, by the way, some treatments for different types of stop codons. How many of you are in genetics? Stop codon, what kind of mutation is that? Yes.
It's a nonsense mutation.
So you have a choice, missense, nonsense. So it is definitely a point mutation forming a stop codon. Agree with your colleague? So this is an example of a point mutation that we'll see very, very frequently where a stop codon gets generated. We actually have drugs like Adalorin, which will allow you to bypass that stop codon. You can actually use this in patients that have cystic fibrosis, where there are some stop codons introduced. And we'll see that sometimes drugs are better than genetic techniques. We'll also talk about antisense oligonucleotides for exon skipping. You can actually show and demonstrate that you can repair, or not repair, bypass truncated exons using exon skipping with oligonucleotides, and we'll talk about this in more detail in the next class. And we can also use these modern techniques, which I am going to highlight in the next class, like CRISPR and Cas9. We have this amazing array of technologies that I want you not to be afraid of learning, not to be afraid of getting some exposure to. And I want you all to do well in this course, but I want you also to be really excited that at some point, all of this is going to mean a lot for you. Whether it's eating yogurt and understanding that bacteria was modified early on, or being able to demonstrate that one day in the very near future, perhaps in Toronto, a gene drive will occur where an entire species or an entire organism will be wiped out because someone said it would be a good idea. That's not as far-fetched as it was a few years ago. And we have the technology now to be able to do all of this pretty much at will. So we'll talk about this more next week. For Thursday, I'm going to ask you to bring the notes that you have. I'll be posting a really short series of notes, and you'll come to the room downstairs, 1085, and we'll go through how to study for the midterm for this particular course.